Yes, so this study was the first global study of the first-in-class EGFR HER3 bispecific antibody iza-bren in patients with metastatic solid tumors, including non-small cell lung cancer. So the study had a dose escalation portion where escalating doses of iza-bren were assessed under 2 different dosing schedules, and that was followed by a dose finding portion where those doses were assessed more closely. And then finally, we're at a dose expansion portion where we are assessing several doses to move forward with 1 dose in the recommended phase 3 dose. Yes, I think this was a long-awaited presentation because we have seen the iza-bren data out of China for a few years now. And so this is the first reports of the global study of iza-bren. And so I think, one, we wanted to confirm, or the audience wanted to know, that the safety profile as well as the efficacy were comparable. And I think that this global study did confirm both, where we're seeing a similar safety profile with no new safety signals, no evidence of ILD. And then regarding efficacy, I think we saw comparable efficacy, especially in certain subgroups, in particular EGFR wild-type non-small cell lung cancer and EGFR mutant lung cancer. Yeah, as we mentioned in our discussion, the patients on Lung 101 were very heavily pretreated. So the range of prior treatments before the study ranged from 1 to 9, but 60% had 3 or more prior lines of therapy. So these are patients that are refractory to treatment, that have very limited standard options left. And so to have a novel first-in-class drug of a novel compound be able to be active in this space and to be tolerated. So, you know, we did see some cytopenias, but really they were manageable and did not really have clinical sequelae. So I think to have something in this space is really critical. And I think there are other tumor types where, you know, after first-line therapy, there are very limited treatments. And so I think beyond chemotherapy and immunotherapy, there are certain disease subgroups that don't have any ADCs or targeted therapies approved. So I think this is a big step forward for a lot of these different cohorts. So we've actually had a lot of different ADC studies at my institution at MSKCC. I have found, you know, using iza-bren to be quite easy. I think the only kind of slight limitation is the day 1, day 8 dosing. So I do see my patients pretty frequently. But besides that, you know, I think cytopenias being the most significant toxicity, again, that's not a toxicity that patients experience clinically. You know, we really haven't had patients need transfusions for their anemia. Again, if there is neutropenia, that's been mitigated with growth factor support. And you know, the rate of treatment discontinuation due to these treatment-related AEs was extremely low at 1.9%. So these are being managed, and you know, patients are continuing on study. The other notable toxicity that we've seen is maybe some fatigue or nausea. And I think with nausea, we have good experience with different antiemetics based on platinum-based chemotherapy. So using dexamethasone, the 5-HT3 antagonist, or other medicines around the time of treatment has been really helpful in decreasing nausea in patients. Yeah, I mean, I think that the space is pretty wide open. You know, we do have datopotamab deruxtecan as you know, with an approval in EGFR mutant lung cancer. But besides that, you know, there's a clinical need for an ADC that is, you know, accessible and available for all comers. After platinum-based treatment with immunotherapy first line, we really have pretty limited options in the second line or beyond. So I do think, one, getting the approval as monotherapy, and then in terms of research opportunities, it's combinations. I think, again, without having significant ILD as a side effect, there is a greater ability to combine with different treatments. And I think the two most interesting things to combine are, with targeted therapy, such as osimertinib, and then with immunotherapy with PD-1 inhibitors. And then I also think that there are, once you have a good antibody, there's also the ability to change out the payload as well, right? So I think, I think this is sort of just the start, and the idea is that we can build on iza-bren monotherapy with these different combinations or different changes in structure.